is present at the time of fertilization determines whether the embryo is going to be a male or a female. In the first 6 weeks, the embryo remains in a sexually indifferent or undifferentiated stage. Okay, in the first 6 weeks, you cannot differentiate whether the embryo is going to be a male or a female. If we could do some genetic analysis, then by looking at the genotype, we might say whether it is going to be a male or a female. But on inspection or by the phenotypic features, we cannot differentiate whether it is a male or a female. So that is during the first 6 weeks. In the 7th week, the indifferent embryo begins some differentiation. Okay, some phenotypic sexual differentiation starts. And by 12 weeks, either male or female characteristics can be recognized externally. The external genitalia can be seen. Okay, so that occurs by week 12. By week 20, by 20th week of gestation, this differentiation is complete. The phenotypic differentiation is complete, and we can differentiate between male and female genitalia. The phenotypic sexual differentiation is determined by the SRY gene located on the short arm of Y chromosome. Okay, if the embryo has Y chromosome, then in its short arm there is a gene called SRY gene. This gene codes for a protein called testis determining factor, or TDF. Okay, and this is the main factor which helps in differentiating the gonads into the male gonad. Okay, the indifferentiated gonad is developed towards the male gonads. Okay. As the indifferent gonad develops into the testis, okay, during its development, there are Leydig cells and Sertoli cells present. Okay, these are the cells present in the testis. These cells differentiate and produce certain factors, like Leydig cell produces testosterone and Sertoli cell produces Mullerian inhibiting factor, or MIF. Okay, Leydig cells secretes testosterone and Sertoli cell secretes MIF. This might come as an MCQ. Okay, in the presence of these factors like testis determining factor, testosterone, and the Mullerian inhibiting factor, the indifferent embryo will be directed towards the male phenotype. The gonad will be a male gonad. But if these factors are absent, that is either the Mullerian inhibiting factor or testosterone or TDF are absent, then the indifferent embryo will be directed towards the female phenotype. Okay, are you getting me?
Yes, sir.
Okay. Okay. In this diagram, you'll see how the undifferentiated gonads develop into either male or the female gonads. Okay. First of all, let us see in this greenish area. Okay. The undifferentiated gonads have a medulla and cortex. Okay. In the medulla, if the genotype of the embryo is XY, that is, it's destined to be a male, then in that case, the short arm of Y chromosome has a SRY gene, and this gene codes for a testis determining factor that helps in differentiation of and development of testis. Okay, testis has Leydig cells and Sertoli cells. Leydig cells secretes testosterone, and Sertoli cells secretes Mullerian inhibiting factor, as I've already told you. Okay, and this testosterone either acts directly on the for the development of the mesonephric duct. Okay, there are mesonephric duct and paramesonephric duct in all embryos. Okay, mesonephric duct is later differentiated into the internal male genitalia if it is stimulated by the presence of testosterone. Okay, and the paramesonephric duct is differentiated into the internal female genitalia. Okay, if there is no inhibiting factor like Mullerian inhibiting factor, then this paramesonephric duct or Mullerian duct differentiates into the internal female genitalia. So basically all human beings are destined to be female. Okay, all of us are destined to be female unless there is a SRY gene that directs us towards the male development. Are you getting me? Okay, so this is the Y chromosome that has the main role. In this greenish area, okay, there is an enzyme called 5-alpha reductase. Okay, this 5-alpha reductase enzyme converts testosterone to dihydrotestosterone, which is 10 times more potent. This dihydrotestosterone acts for the development of the external male genitalia, okay, like penis, scrotum, and prostate. Now, let us look at the pink area. Okay, In those with XX genotype, there is no SRI gene. So there will not be development of testes, but there will be development of the ovaries by default. Okay, ovaries secrete the hormone estradiol. This hormone has a role in developing the external female genitalia. Okay, like clitoris, labia, and distal vagina. Okay, and the paramesomorphic duct or the Mullerian duct by default develop into the internal female genitalia that includes the fallopian tubes, uterus, and the proximal vagina. Okay, and in this diagram you can see there are two numbers, 1 and 2 written. Okay, these are written in order to see what happens if these factors are absent. Okay, can anyone guess what would happen if there is absence of Sertoli cells or the Sertoli cells do not secrete Mullerian inhibiting factor? Can anyone guess what would be the genital tract of that individual? How would that be? Anyone? Or do you want me to ask one of you by name? Anyone wants to volunteer? And it doesn't matter if you are wrong, okay? Anyone, just make a guess. कसले भने प्रकृतिले नै भन नि त
थाहा भएन
ल यसमा चाहिँ के हुन्छ भन्दाखेरि तिम्रो सर्टोली सेल्स भएन भनेदेखि तिम्रो मुलेरियन इन्हिबिटिङ फ्याक्टर नहुने भयो, राइट? एमआइएफ नहुने बित्तिकै तिम्रो प्यारामेसोमेफ्रिक डक्टबाट इन्टरनल फिमेल जेनेटिलाई हुने प्रोसेसलाई इनिबिट गर्ने फ्याक्टर भएन नि त, भनेपछि यो इनिबिसन नहुने भयो, बुझ्यौ? बेसिकली त्यो इन्डिभिजुअलको सर्टोली सेल्स छैन र मुलेरियन इन्हिबिटिङ फ्याक्टर छैन भन्नुको मतलब त्यो पेसेन्ट मेल वाइ क्रोमोजोम त छ उसको, हैन? एक्सवाई क्रोमोजोम भएको मान्छे हो त्यो एम्ब्रियोमा चाहिँ यदि एक्सवाई क्रोमोजोम छ भने एसआरआई जिन हुने भयो, त्यो जिनले टेस्टोस्टेरोन फ्याक्टर निकाल्ने भयो, त्यसले टेस्टिसको डेभलपमेन्ट गराउने भयो। तर टेस्टिसको डेभलपमेन्ट भए पनि सर्टोली सेल्स भएन अथवा एमआइएफ भएन भने के हुन्छ भनेर मैले सोधेको नि त्यसमा, हैन? त्यही भएर टेस्टिस छ भन्नुको मतलब लिडिक सेलबाट टेस्टोस्टेरोन आउँछ, त्यसले चाहिँ डायरोटेस्टोस्टेरोन पनि रिलिज गर्छ, जसले गर्दा एक्सटर्नल मेल जेनेटिलाई हुने भयो। टेस्टोस्टेरोन सेक्रेट भएकै हुन्छ, त्यही भएर मेसोनेफ्रिक डक्टर डेभलपमेन्ट भएर इन्टरनल मेल जेनेटिलाई हुने भयो। अनि इन्टरनल मेल जेनेटिल र एक्सटर्नल मेल जेनेटिल चाहिँ हुने भयो त्यो पेसेन्टको, त्यो मान्छेको, हैन? तर तिम्रो सर्टोली सेल्स र मुलेरियन इन्हिबिटिङ फ्याक्टर नभएको कारणले गर्दाखेरि तिम्रो प्यारामेसोनेफ्रिक डक्ट पनि डेभलप हुने भयो क्या अब, बुझ्यौ? देयर विल नट बी इनिबिसन अफ दिस प्यारामेसोनेफ्रिक डक्ट, सो द इन्डिभिजुअल विल अल्सो ह्याभ इन्टरनल फिमेल जेनेटिलिया, fallopian tubes, uterus, proximal vagina हुन्छ। रुडिमेन्ट्री फर्ममै हुन सक्छ, बट द्याट विल बी प्रेजेन्ट, बुझ्यौ? एउटै इन्डिभिजुअलमा
एक्सटर्नली चाहिँ कहाँ ओपन हुन्छ त?
एक्सटर्नल ओपनिङ हुँदैन, एक्सटर्नल त्यही भएर त प्रोक्सिमल भजाना भनेछ नि, तिम्रो युटरस प्रोक्सिमल भजाना कलर डिस्टल ओपनिङ त नहुने भयो नि त, हैन? किनभने यसको डेभलपमेन्ट हामीले एकछिनमा हेर्छौँ डिस्टल भजाना केले बनाउँछ भनेर, हैन? तर यो इन्डिभिजुअलमा चाहिँ उसको एक्सटर्नल र इन्टरनल जेनेटिलिया चाहिँ मेल चाहिँ हुने नै भयो, प्लस फिमेलको पनि सम पार्ट्स हुने भयो, फलोपियन ट्युब्स, युटरस, प्रोक्सिमल भजानाको रुडिमेन्ट्री फर्म हुन सक्ने भयो, बुझ्यौ? त्यो चाहिँ सर्टोली सेल्सको एब्सेन्ट भयो भने। सपोज अब 5 अल्फा रिडक्टेज भन्दा इन्जाइम डेफिसिएन्सी भयो भने के हुन्छ होला त? त्यो भनेको मतलब त्यो पेसेन्टको त्यो मान्छेको पनि जेनोटाइप चाहिँ एक्सवाई नै हो, एक्सवाई भएपछि टेस्टिस हुने भयो, टेस्टोस्टेरोन सेक्रेट हुने भयो। तर टेस्टोस्टेरोनबाट डायरोटेस्टोस्टेरोन भन्ने पोटेन्ट फर्ममा कन्भर्ट गर्ने इन्जाइम 5 अल्फा रिडक्टेज नभएपछि तिम्रो एक्सटर्नल मेल जेनेटिलियाको डेभलपमेन्ट चाहिँ नहुने भयो, ओके न? 5 अल्फा रिडक्टेज भन्ने इन्जाइमको डेफिसिएन्सीले गर्दाखेरि। तर यो जुन डेभलपमेन्ट नहुने भन्ने कुरा चाहिँ कहिलेसम्म हुन्छ भने प्युबर्टीसम्म मात्रै हुन्छ। प्युबर्टीसम्म त्यो टेस्टोस्टेरोन हर्मोनको लेभल कम हुन्छ, त्यो कारणले अन्टिल प्युबर्टी चाहिँ त्यो मान्छेको चाहिँ एक्सटर्नल जेनेटिलिया एम्बिगस जस्तो देखिन्छ, हैन? वी क्यानट डिफरेन्सिएट वेदर इट इज मेल अर फिमेल, एम्बिगस जेनेटिलिया हुने भयो। तर तिम्रो प्युबर्टी भइसकेपछि चाहिँ टेस्टोस्टेरोनको हर्मोनको लेभल नै यति धेरै बढ्छ कि त्यही टेस्टोस्टेरोनले नै एक्ट गरेर एक्सटर्नल मेल जेनेटिल डेभलप हुन्छ, बुझ्यौ? ल अघि मैले सोधेको क्वेसनको एन्सर यही हो है त। नम्बर 1 मा चाहिँ यदि इफ देयर इज नो सर्टोली सेल्स और ल्याक अफ मुलेरियन इन्हिबिटिङ फ्याक्टर देन दे विल डेभलप बोथ मेल एन्ड फिमेल इन्टरनल जेनेटिलिया एन्ड मेल एक्सटर्नल जेनेटिलिया, right? And if there is 5 alpha reductase deficiency, then there is inability to convert testosterone to dihydrotestosterone, टेस्टोस्टेरोनबाट आएर टेस्टोस्टेरोन नबन्ने भयो. सो द मेल इन्टरनल जेनेटेलिया चाहिँ राम्रोसँग डेभलप, मेल इन्टरनल जेनेटेलिया चाहिँ डेभलप हुने भयो. तर देयर विल बी एम्बिगस एक्सटर्नल जेनेटेलिया, एम्बिगस एक्सटर्नल जेनेटेलिया अन्टिल प्युबर्टी. किनभने प्युबर्टी पछि चाहिँ देयर विल बी इन्क्रिज इन टेस्टोस्टेरोन लेभल द्याट कज मास्कुलिनाइजेसन. यसको चाहिँ मास्कुलिनाइजेसन गराएर तिम्रो मेल एक्सटर्नल जेनेटेलिया चाहिँ डेभलप हुन्छ है त. अब यो चाहिँ नेमोनिक छ, यो मैले फिगर चाहिँ फर्स्ट फर युएसएमएली भन्ने किताबबाट लिएको स्टेप 1 को. यसमा लिडिक सेल्स, लिडिक लिड्स टु द मेल sexual differentiation, both internal and external, and Sertoli shuts down female. Okay, the Sertoli has secretes, cell secretes mullerian inhibiting factor that inhibits paramesonephric duct. So this shuts down, Sertoli shuts down female internal sexual differentiation. Right? Everyone is clear?
What's this, sir?
Okay. Now let us go to the development of the gonads. Okay, the ovary. First of all, you have to know is that the gonads develop from the mesoderm. Okay, the intermediate mesoderm forms a longitudinal elevation along the dorsal body wall. Okay, along the dorsal body wall, there will be a longitudinal elevation formed that is called as urogenital ridge, and this urogenital ridge later forms the gonadal ridge, and from this gonadal ridge, the gonads get developed. The primary sex cords develop from the gonadal ridge and incorporate primordial germ cells that have the XX genotype, which migrate into the gonad from the wall of the yolk sac. From the wall of the yolk sac, the primary sex cords develop and that incorporate the primordial germ cells. Okay, the primary sex cords extend into the medulla and develop into the rete ovary, which eventually degenerates. This rete ovary degenerates later on, but from the wall of the yolk sac, there will be migration of the primordial germ cells into the gonadal ridge, and that later develop the ovary. Okay, the secondary sex cords develop and incorporate the primordial germ cells. Around the primordial germ cell, there will be formation of the secondary sex cords, and that will form the thin tunica albuginea. The secondary sex cords later break down. Okay, they break down and form isolated cell clusters. There will be a small group of cells. These are known as the primordial follicles, and these contain the primary oocytes, which are surrounded by simple squamous cells. Now, during the formation of the ovaries, they are actually in the abdominal region. Okay, the ovaries originally developed within the abdomen, but later they undergo a relative descent. Okay, this descent is called relative because this is not actual migration of the ovaries, but there is a disproportionate growth of the embryo. Because of the disproportionate growth, the abdominal area grows faster, the pelvic area grows slower. Okay, so it seems like there is a relative descent of the gonads into the pelvis. Okay, and this descent may also be explained by the presence of gubernaculum. Okay, the gubernaculum is a band of fibrous tissue along the posterior wall of the uterus that extends from the medial pole of the ovary to the uterus. Okay, have you heard about the ovarian ligament? Ovarian ligament joins the ovary with the uterus, right? The medial portion of the uterus and the medial portion of the ovaries are joined together by the ovarian ligament. This ovarian ligament is formed by the gubernaculum. Okay, this gubernaculum also extends further. This continues into the labia majora and later on form the round ligament of the uterus. So both the round ligament of the uterus and the ovarian ligament are formed from the gubernaculum. And because of the presence of this gubernaculum, it is hypothesized that there is descent of the gonads, descent of the ovary into the pelvis. Okay, that is one possible explanation, and another explanation is the disproportionate growth of the abdomen. Now let us go to the development of the paramesonephric duct. What does the paramesonephric duct do? Okay, paramesonephric duct is also known as Mullerian duct. The cranial portions of the paramesonephric duct develop into the uterine tubes. Okay, this is the main ducts that forms the female internal genitalia. Okay, the paramesonephric duct. The caudal portion of the paramesonephric duct will fuse in the midline and form the uterovaginal primordium, while the cranial portion of the paramesonephric duct develop into the uterine tubes or the fallopian tubes. Okay, I'll show you the diagram in the next picture. In the next slide, the uterovaginal primordium develops into the uterus, cervix, and the superior one-third of the vagina. From the paramesonephric duct, the cranial portion will become the fallopian tubes. The mid portion will form the uterovaginal primordium after the two tubes fuse together, and this gives rise to the development of the uterus, cervix, and the upper one-third of the vagina. The paramezonephic ducts project into the dorsal wall of the cloaca. Do you know what cloaca is? It is an area in the distal end of the embryo, okay, where the urogenital sinus and the alimentary tract or the GI tract open. Okay, they are together, fused together in the cloaca. In the dorsal wall of the cloaca, there will be the projection, there will be some sort of projection of the paramezonephic duct, and this will induce the formation of the sinovaginal bulbs. The sinovaginal bulbs will later form the solid vaginal plate that will get canalized and develop into the lower two-third of the vagina. Okay, so the lower two-third of the vagina is formed by the sinovaginal bulb, while the upper one-third of the vagina, uterus, and the fallopian tubes are formed from the paramezonephic duct. Okay, the vestigial remnants of the paramezonephic duct may be found in the adult female and are called the hydatid of Morgagni. Even the paramezonephic duct will have some remnants, even in females. Okay, in females, paramezonephic duct is well developed. However, there will be some vestigial remnants of its proximal portion, and that is known as the hydatid of Morgagni. Okay, now here's the diagram. Here you can see this is the mesonephros. Okay, this is the mesonephric duct on either sides. This is the alimentary tract or the GI tract. This is the cloaca. Okay, and here this is the connection of the bladder. Okay, this cloaca will later on form the bladder. On either sides of the mesonephros, there will be formation of the paramesonephric duct. Can you see the pinkish colored tubes on either sides of the mesonephros? Okay, these are the Mullerian ducts or the paramesonephric ducts. Okay, the paramesonephric ducts will later on form the tubes that will get fused in the midline and will form the uterus and the fallopian tubes. And if you see the development of the mesonephros and the metanephros, from one end of the mesonephric duct, there will be origin of ureteric bud and metanephros, and that will later form the kidneys. Okay, here in this diagram you can see from the metanephros there is formation of kidney. Now if you see the another diagram, okay, now you can see the pinkish colored is the Mullerian duct or the paramesonephric duct. Okay, the two paramesonephric ducts on either sides get fused in the midline, right? Get fused in the midline. This is the fused Mullerian ducts, and this will later form the uterus. Fused portion forms the uterus, and the unfused portion or the unfused proximal portion form the two fallopian tubes on either sides. Okay, can you make out in the diagram? And at a place where the fused paramesonephric duct joins to the cloaca, there will be formation of the sinovaginal bulb. It's not shown in the diagram, and that will later form the lower two-third of the vagina. Okay, I'll show you another diagram. In this diagram, you can see there are two paramesonephric ducts on either sides of the mesonephros. Okay, these two paramesonephric duct will go on developing and then later fuse in the midline. Okay, you can see in this diagram there is fusion of the two paramesonephric ducts. After fusion, this will form the uterus, cervix, and upper one-third of the vagina. And at a place where the fused paramesonephric duct joined to the urogenital sinus, okay, there will be formation of the vaginal plate, sinovaginal bulb, and from the sinovaginal bulb, the development of the vaginal plate that will later get canalized and form the lower two-third of the vagina. Okay. Now, in this diagram, you can see the different structures that give rise to the development of the female gonads. Okay, there is a urogenital ridge. You can see two pair of tubes. Okay, one is the mesonephric duct with mesonephric tubules. Okay, the two mesonephric ducts and the two mesonephric tubules, and there is a paramesonephric duct on either sides. The two paramesonephric ducts on either side get fused in the midline to form the uterovaginal primordium that will form the uterus, cervix, and upper one-third of vagina. And on the inferior aspect of the fused uterovaginal primordium, there will be the formation of the sinovaginal bulb that will later get differentiated into the vaginal plate that gets canalized and form the lower two-third of the vagina. Now, in this diagram, you can see the different vestigial remnants. Okay, this is a normal, this is the uterus, two tubes on either sides. Hydatida of Morgagni is a remnant of vestigial remnant of the paramesonephric duct, because see, you can see the paramesonephric duct forms the fallopian tubes. So Hydatida of Morgagni is the vestigial remnant of the paramesonephric duct. Similarly, you can see the dotted tubes on either sides of the uterus. Okay, this is the, what is this? This is the, what is this? Anyone? This is the mesonephric duct. Okay, the mesonephric duct, proximal portion will have the vestigial remnant of appendix vesiculosa, hypophoron, paraphoron. These are all the vestigial remnants of the mesonephric duct. Okay, and the mesonephric duct on the lower side may lead to the formation of Gartner's cyst. Okay, the duct of Gartner, the Gartner's duct is a vestigial remnant of the mesonephric duct. In pathological conditions, some of the females may come with the problem of a cystic lesion in the vagina. Okay, and sometimes that cystic lesion is nothing but the cyst of Gartner's duct cyst that forms because of collection of some fluid within that duct. Okay, and I had told you about the gubernaculum, right? The ovary is attached to the medial portion of the uterus by the ovarian ligament. Okay, this ovarian ligament is formed from the gubernaculum. You can see here, this is the ovarian ligament. And there is also the continuation of the ovarian ligament or the gubernaculum into the labia majora, okay, through the round ligament. This is the round ligament on either sides. Okay, are you getting me?
Yes, sir.
Okay, and in the diagram, you can see the upper 1/3 of the vagina is formed from the paramesonephric duct, while the lower 2/3 is formed, or the inferior 2/3 of the vagina is formed by the sinovaginal bulb. Now let us see what the mesonephric ducts will lead to. Okay, the mesonephric ducts develop in the female as part of the urinary system. Okay, mesonephric ducts will form the urinary system. These are critical in the formation of the definitive metanephric kidney. Okay, in the last diagram, I showed you how the distal end of the mesonephric duct led to the differentiation into the metanephric kidney. Okay, however, the mesonephric ducts will later degenerate after formation of the metanephric kidney. The vestigial remnants of the mesonephric ducts may be found in the adult female called the appendix vesiculosa and Gartner's duct that I just showed in this diagram. Okay, appendix vesiculosa and the Gartner's duct. These are the vestigial remnants of the mesonephric ducts in female. And vestigial remnants of the mesonephric tubules, also called as epiphoron and paraphoron, may be found in the adult female. Okay, this epiphoron and paraphoron are nothing but small cyst-like lesions that you can see when we operate on the pelvis. For example, while we are doing cesarean section or during any pelvic surgery, if we see on either sides of the fallopian tubes, we can see small cyst-like lesions on the walls of the fallopian tube in the broad ligament. Okay, these are the epiphoron and paraphoron. These are not pathological, but these are just the vestigial remnants of the mesonephric tubules. Okay. Now in this diagram, I have kept to show you how the sinovaginal bulb is formed. Okay. In figure A, you can see that the two paramesonephric ducts of the two Mullerian ducts are fused. Okay, these fused Mullerian ducts join the urogenital sinus. This is the urogenital sinus. These fused Mullerian ducts fuse with the urogenital sinus at the Muller's tubercle. Okay, this area is called the Muller's tubercle, right? And from the urogenital sinus, the sinovaginal bulbs evaginate and proliferate cranially. From the urogenital sinus, okay, there will be the formation of sinovaginal bulb. The sinovaginal bulbs evaginate and proliferate cranially to create the vaginal plate. This will form the vaginal plate. You can see in this diagram. This vaginal plate is later canalized. Okay, and after canalization, this will form the distal 2/3 of the vagina. Okay. Now, how are the external female genital developed? Okay. Until now, we are discussing about the development of internal female genitalia. Now, for the development of the female external genitalia, the primordia comes from the mesoderm. Okay, a proliferation of mesoderm around the cloacal membrane. When the mesoderm around the cloacal membrane proliferates, there will be rise of overlying ectoderm. The ectoderm that lies just above the mesoderm will rise up, and there will be formation of three distinct structures. Okay, that are the phallus, urogenital folds, and the labioscrotal swellings. Okay, three distinct structures are visible externally when there is proliferation of the mesoderm near the cloacal membrane that leads to the overlying ectoderm to rise up. Okay, and all of these three structures lead to development of certain structures in adults. For example, the phallus will form the clitoris, that includes the glans clitoris, corpora cavernosa clitoris, and the vestibular valves. Okay, basically the phallus forms the clitoris, the urogenital folds will form the labia minora, and the labioscrotal swellings will later differentiate into the development of labia majora and mons pubis. Okay, in this diagram you can see the three structures. Okay, this is the phallus, this is the urogenital folds, and there is a labioscrotal swelling on either sides of the urogenital folds. Okay, this diagram is at 5 weeks of gestation. At 5 weeks of gestation, the embryo has these structures. At birth, you can see that the phallus differentiates into the clitoris. Okay, this is at birth. At the clitoris, the urogenital folds will form the labia minora, and the labioscrotal swellings on either sides of the urogenital folds will form the labia majora and the mons pubis. Okay, in figure C, you can see this is the diagram of the female external genitalia at birth. Okay, there are two labioscrotal swellings that form the labia majora. There is labia minora in between, and the clitoris that is differentiated from the phallus. Okay, now this slide is important because you can get any MCQ from this slide. Okay, many MCQs can be made. This diagram shows the vestigial remnants of different organs of the female genital tract. Okay, number one is hydatid cyst of Morgagni. Hydatid cyst of Morgagni is the vestigial remnant of paramesonephric duct. Similarly, number two is the Cobell cyst. Cobell cyst is formed from the appendix vesiculosa, which is the vestigial remnant of the mesonephric duct. Then there are cyst of Epiphoron and the cyst of Paraphoron, 3 and 4. Okay, these are formed from the vestigial remnants of the mesonephric tubules. And number 5 is the Gartner's duct cyst. Okay, can you see the cyst here? Okay, this is the Gartner's duct, and there is a cyst formed in the Gartner's duct. This Gartner's duct cyst is formed by the vestigial remnant of the mesonephric duct. Okay. Now let's go to some clinical considerations. Okay, why are we studying about the development of the female genital tract? We are studying because we need to know how the normal organs develop and what happens if there is any abnormality or any defect in the process of this development. Okay, the uterine anomalies have been classified into 7 classes. Okay, class 1 to class 7. And class 1 includes Mullerian hypoplasia or agenesis. Class 1 is agenesis. Okay, so agenesis can occur for any part of the paramesonephric duct. If there is agenesis of the proximal portion, then there will be no fallopian tubes. For example, in figure 4, you can see the uterine tubes are absent. There is a uterus, there is a vagina, there is a cervix, but you cannot see the tubes on either sides. So this is uterine tube agenesis. Similarly, in number 3, you can see the fallopian tubes are intact, the vagina is intact, but the uterus and cervix, this is hypoplasia. Okay, there is uterus and cervix hypoplasia. In number 2, you can see that there are 2 tubes well developed, fallopian tubes are normal, uterus is normal, vagina is normal, but there is no development of the cervix. Okay, so this is cervical agenesis. In number 1, you can see the tubes are normal, the uterus is normal, cervix is normal, and vagina is normal up to the upper 1/3, but there is no communication to the exterior. So this is lower vaginal agenesis. Okay, lower vaginal agenesis is one of the cause of primary amenorrhea. Okay, when a female reaches puberty at around 11 to 13 years of age, usually females start menstruating. But if there is no lower vagina, then there will be no outflow tract. So the female will present with severe abdominal pain that is cyclical during the menses, as there is collection of fluid within the collection of blood within the uterus and the upper one third of the vagina. So there will be hematocolpus, there will be hematocolpus is the collection of blood in the uterus. The colpus means collection of blood in the vagina. So there is collection of blood in the uterus and the vagina. So that is known as hematocolpus. Okay, that may be presented as a mass in pelvis. On abdominal palpation, you may feel a pelvic mass. If a 13-year-old female who presents with cyclical pain abdomen, there is no menses, then you have to suspect there might be some agenesis or some defect in the outflow tract of the female genital tract. Okay, so this is about the class 1 anomaly of the uterus, also known as Mullerian hypoplasia or agenesis. Next is class 2. Okay, class 2 is unicornate uterus. At the end of this class, I'll tell you about mnemonics that you can use to remember these abnormalities. Okay, so first is agenesis. Second is unicornate uterus. Unicornate uterus means there will be just one horn. Okay, one arm of the the paramesonephric duct will be well developed, while the other part of the paramesonephric duct or the other half of the paramesonephric duct is incompletely developed or fails to develop. Okay, so you can see in this diagram in number 1, the right side of the fallopian tube is normal and there is uterus, but the left half of the fallopian tube is not well developed. Okay, this is incompletely developed. So this is unicornate uterus with a communicating rudimentary horn. Rudimentary horn means the incompletely developed fallopian tube, incompletely developed paramesonephric duct. Okay, this rudimentary horn has communication. In this diagram, you can see there is a communication between this cavity and the uterine cavity, main uterine cavity. So this is the communicating rudimentary horn. Unicornate uterus with a communicating rudimentary horn. And number 2, this is the same picture as in number 2. Okay, there was a mistake in this diagram. They try to show unicornate uterus with a rudimentary horn that is non-communicating. So there had to be some blockage over here. Okay, but this is not shown over here. This diagram is wrong. And number 3, you can see unicornate uterus with a rudimentary horn that has no cavity. Okay, this rudimentary horn has no uterine cavity. So there is no endometrial tissue. There will be no endometrial proliferation during menstruation. So there will be no bleeding. But in case of unicornate uterus with the rudimentary horn with cavity, they will have some endometrial tissue. So there will be some bleeding from this endometrial tissue as well. If there is communication, that blood will get drained. But if there is no communication, then that blood will get accumulated over different cycles. So there will be formation of a swelling on one side of the uterus. Okay, so this will be a pathological condition. Third is uterine didelphys. Okay, uterine didelphys. Didelphys means double uterus. So there will be 2 uterus. This occurs if there is a complete lack of fusion of the paramesonephric duct. In the 2 paramesonephric ducts do not fuse with each other at all. Okay, if they do not come near the midline, then what happens? The one side of the paramesonephric duct will form its one tube, its one ovary, it's one tube and its one uterus, and the other side of the paramesonephric duct will also do the same thing. So there will be 2 fallopian tubes. Okay, in this diagram you can see there are 2 fallopian tubes, there are 2 uterine cavities. Okay, and then there is the vagina. In number 1, you can see this is uterine didelphys with normal vagina. There is a single vagina, but there are 2 uterus and there are 2 fallopian tubes. Okay, one from each side. In number 2, you can see this is the didelphys with complete vaginal septum. Okay, the 2 parts or the 2 paramesonephric ducts are fused only in the distal portion, only in the lower third. So there will be some formation of vaginal septum. Okay, in this diagram, this is the HSG. HSG means hysterosalpingogram. In this procedure, we push a dye from the vagina into the uterine cavity and then take an X-ray to see how the filling defect is. Okay, you can see there are two tubes on either sides. Okay, this is one uterus, this is another uterus. Okay, and then there are two separate vagina, and there is a septum in between. Okay, this is the diagram for this. As I say, picture of this diagram, number 2. Okay, are all of you getting me? If you have any questions, you can ask. Okay, shall I continue?
Yes, sir.
Okay. Next is bicoordinate uterus. Okay, class 4 anomaly is the bicoordinate uterus. Bicoordinate means there will be partial fusion of the paramesonephric duct. The two paramesonephric ducts will get partially fused. So there will not be two separate uterus, but there will be some partition in between the two cavities. Okay. So in number 1, you can see bicoordinate uterus with a complete division down to the internal os. So there are two separate uterus, but the cervix is single. Okay, in number 2, you can see the bicoordinate uterus with partial division. There is only partial division. So there is a septa in between the two cavities. Okay, this is the HST diagram of the bicoordinate uterus. You can see the two horns. Number 5 is the septate uterus. Okay, septate uterus occurs when the medial walls of the caudal portion of the paramesonephric ducts partially or completely fail to reabsorb. What happens is when the two paramesonephric ducts come together, they fuse with each other. And then after fusion, the wall in between the two tubes will get absorbed. Okay, so there will be formation of a single cavity. But if the wall between the two paramesonephric ducts do not get fully resorbed, then there will be the formation of septum. Okay, in number 1, this is a septate uterus with complete septum down to the external os. Okay, you can see the septa from the fundus to the cervix. Okay, this is the septate uterus with complete septum, and here is in number 2, this is septate uterus with partial septum. Okay, the septum hasn't reached up to the os. This is class 5 anomaly. Next is class 6 anomaly is arcuate uterus. Okay, arcuate uterus is just a mild deviation. Okay, in this type of uterine anomaly, there will be a small dimpling at the level of the fundus. Okay, the anatomical hallmarks include a slight midline septum within a broad fundus, sometimes with minimal fundal cavity indentation. So such type of findings we can encounter during cesarean section, where there will be some arc-like structure. Okay, there will be arc at the level of the fundus. And most clinicians report no impact on reproductive outcomes. However, there was one study done by Ulfa and colleagues, and who found that those women with arcuate uterus have excessive second trimester losses and preterm labor. Okay, so the findings are contrasting. In this diagram, you can see this is the normal uterus. Okay, you can see a well-developed fundus, but here the fundus is not that developed. Okay, there is a small dimpling. Okay, this is not very clear cut, but there is a small dimpling, and this is the arcuate uterus. There will be no any anomaly inside. Okay, inside will be same. Okay, and the last is the diethylstilbestrol induced reproductive tract abnormalities. Okay, DES induced reproductive tract abnormalities. Okay, diethylstilbestrol is a non-steroidal estrogen. Okay, this was used for supporting pregnancy because it has some effect of estrogen. It was used in the past for supporting pregnancy for patients with recurrent miscarriages, okay, for patients with scrotal cancer, breast CA. Okay, this was used. But later it was found that those women who were exposed to the diethylstilbestrol, they later on developed, the child of those women developed clear cell adenocarcinoma of the vagina and cervix. Okay, and even the female childs of those women were found to have the T-shaped uterus. So this was later on found that this drug is teratogenic, so people stopped using this drug. Okay, this drug is no longer used in obstetrics. However, in case and It reads as A, you don't be sad. Okay. A, you don't be sad. A is agenesis. U is unicornuate uterus. Don't stand for didelphys. Okay. B means bicornuate. And sad, S is for septate. A is for arcuate. D is for de-escalated. Okay. So there are class 1 to class 7 uterine anomalies. And A, you don't be sad. Agenesis, unicornuate, didelphys, bicornuate, and SAD stands for septate, arcuate, and de-escalated anomalies. Okay. Now we have only one more slide. Okay, the ovarian anomalies. Sometimes we can encounter some ovarian anomalies. The terms are supernumerary ovary. Supernumerary ovary means there are presence of ovary or ectopic ovary that has no connection with the broad ligament, uterovarian ligament, or the inferior oblique ligament. Okay, these ligaments are all the ligaments that lie in the pelvis and connect the uterus and ovaries with the lateral pelvic wall. Okay, the broad ligament, uterovarian ligament, and infundibulopelvic pelvic ligaments. If the ectopic ovarian tissue has no connection with these ligaments, then this is a supernumerary ovary, the case of supernumerary ovary. However, if there is an excess ovarian tissue nearby the normal ovary and is connected to the normal ovary, then that is known as accessory ovary. Okay, supernumerary ovary means it is arising at a different site, at the ectopic site, while accessory ovary means the excess ovarian tissue is nearby and connected to the normally placed ovary. And sometimes the ovaries may be absent. Okay, and these absent ovaries may be because of the congenital agenesis or when there is ovarian torsion, which later on go into necrosis and are reabsorbed. Okay, when there is ovarian torsion, there will be some necrosis and the ovarian tissue may get reabsorbed, and later on investigation, we may not find any ovary. That is known as the absent ovary. Okay, thank you. So this is the end of my class. So if you have any questions, please you can ask me.
Not you.